Thank you for joining us at the ASM Distinguished Lecturer virtual series for the ASM student and postdoc chapters. We really appreciate you joining us, whether it's morning or evening or even overnight for you. We're happy to have you here. This series of seminars is designed to support some of the most important ASM members, and that is you, the early career scientists who are involved in roughly 150 ASM student and postdoc chapters located throughout the US and the world. I'm Brianna Burton. I'm the chair of the ASM Distinguished Lecturer Committee, and I'm a faculty member in the Department of Bacteriology at the University of Wisconsin in Madison. For those of you who are not familiar with the ASM Distinguished Lecturer Program, this program provides keynote speakers once a year to each of the ASM branches. And we select a roster of speakers whose expertise is wide-ranging across areas of microbiology. And the branches then choose a speaker who meets their programmatic needs. And this is one important way that ASM National supports local branches and their members. The program has been ongoing for over 50 years because of the great success and the fantastic speakers we have. And you'll hear from one who I'll introduce in just a moment. But first, ASM has asked me to share a couple of slides with you about resources for ASM members. And so moving on to the next slide, whether you are part of a student or postdoc chapter or a local branch, the parent organization, ASM National, provides additional value through the community for its members. Oh, thanks, Anne. And would like to make sure that you're aware that in addition to being a member of a branch, you can also be a member of the ASM National, and you can find more information about what ASM National provides at that website. And other direction. Thank you. And then just two examples of some of the resources they provide. This is one of them. It's called ASM Connect. It's an online network for discussion and an open forum where you can connect to other ASM members. It's great for getting advice, asking about resources or tools, and learning about different areas of microbiology in the community. And the last thing I'll share with you, another resource that ASM provides is through the salary survey that they conduct. And they provide the information collected from that survey. And you can, as an ASM member, gain access to this to learn about what you can expect for compensation and benefits in your field. It can help you negotiate salary when applying for jobs. So I encourage you to check out this resource as well. So turning to the reason that we're all here today, I'm excited to introduce Dr. Jason Bartz. He's a professor of medical microbiology and immunology at Creighton University in Omaha, Nebraska. And he completed his bachelor's in biology with a minor in chemistry at the University of Wisconsin at Stevens Point. And then moved on to the University of Wisconsin at Madison, where he eventually completed his Ph.D. in the veterinary science program. And as you'll hear shortly, his research is focused on one of the most challenging areas of investigation in infectious disease biology, and that is that of infectious protein transmission. But beyond the research in his lab that he'll tell you more about, he's served on editorial boards of journals, including PLOS Pathogens and Journal of Virology. He's been on the board of scientific counselors at the NIAID at NIH. But I think what's really important in terms of people who we have in this ASMDL roster is what one of the nominators of Dr. Bartz said when nominating him for this lectureship. And that person noted, quote, "Dr. Bartz exemplifies a spirit of cooperation and kindness," unquote. And they went on to explain that it's really his leading by example in this way that has been instrumental in building community in his department and institution and making it a supportive and productive environment for trainees. And so in addition to accolades about science and science communication, I think that is something that is really important and we look for in people who we look to as leaders in the field. And before I turn this over to him, I just want to mention also that he has a long list of community volunteering as well, including serving as a science fair judge and staff photographer for local schools. And related to that, he has a longstanding interest in photography. And if it's all right, we will share the link to the photography website for anyone who's interested to take a look at that. One last note that I'll make for all of our listeners, before Dr. Bartz begins, I will remind you that in this era of Zoom where most of the cameras are off and our speaker cannot see you, the best way to thank him is for you to ask great questions at the end of the seminar. You can actually enter these into the chat window at any time during the seminar. So please keep yourself muted, write your questions into the chat. Include your name and your institution with your question, and I will read these out to Dr. Bartz at the end of the seminar. And so with that, I'm going to mute myself and turn this over. Please welcome Dr. Bartz.
Thank you very much. So thank you for the very kind introduction. And what I want to talk about today are prions, and this is an overview of what the areas I want to cover today. So first I want to talk a little bit about the agent. So as was alluded to in the introduction, is that this is kind of the neither world of microbiology, these infectious proteins. And I want to talk about the prion agent and kind of how it's unique in biology, how if you are a protein-only infectious agent that you can make more of yourself. Some of the consequences of being a protein-only agent as far as the physical properties. Talk a little bit about prion strains. This is kind of related to CWD, and then the diseases that are caused by prions. And then get into the main part of the talk on chronic wasting disease. And I kind of decided, instead of giving, more like a didactic kind of lecture thing about CWD, is I wanted to put this into historical context and really kind of emphasize to everybody here how research builds upon each other. And specifically with chronic wasting disease, it's really intertwined with our knowledge and on basic science on prions. And I want to kind of emphasize that. So as I go through the 1980s, 1990s, and 2000s, what I'm going to do is I'll have 2 slides on chronic wasting disease, which I think were some major findings, and then 1 slide on prion diseases, and really show how this work really built upon each other, and sometimes in really kind of unexpected ways. All right, so I'll talk about the prion agent. As mentioned before, this is unique in biology. It's infectious protein, but it's long been recognized that prions had really unusual properties. So back in the 1960s, Tika Alper did some really seminal work in the field, and what she found is that prions were unusually resistant to inactivation by irradiation. And so scrapie, and I'll talk about this in a few slides, this is a prion disease of sheep and goats. And the agent that caused this disease, which was unknown at the time, was extraordinarily resistant to inactivation by irradiation. And kind of consequent with this is in 1967, a mathematician named Griffith proposed this idea of a protein-only infectious agent. So this kind of theoretical underpinning then led to Stan Prusner's work back in 1980, and I'm going to go through a few of these slides later on, but really this Herculean effort. And what he showed is that these amyloid deposits that are in all prion-infected animals, if you purify those, they're composed of these long, linear, non-branching amyloid fibrils, and that these fibrils are mainly comprised of the single protein called the prion protein. And this hypothesis, the prion hypothesis by Stan Prusner in 1982, stated that this protein was the infectious agent, which at the time was very highly controversial. It wasn't until 2007 that a former postdoc of Prusner's, Serge Isopatapone, actually, in my opinion, did the definitive experiment where he's able to reconstitute prion infectivity from non-infectious components. So in 1997, Stan Prusiner received the Nobel Prize for his work on prion diseases. So if you're a protein-only infectious agent, so this is really a kind of a disease of protein conformation. And this prion protein that Prusiner identified in 1982 exists in two distinct forms. PRPC, and the C stands for cellular. This is the normal form of the protein that's in all of us right now. It has a high alpha helical content. Pictured down here is the NMR structure of it. Sensitive to proteases, soluble detergents, not infectious, and the function is really unclear. There's a lot of work going on in the function of PRPC, but it's really not known. So through a post-translational conformational change, PRPC changes into PRPSC. This is the infectious self-templating form of the protein. It's resistant, highly resistant to protease K digestion, pictured here in a Western blot, showing you with protease K or thermolysin. This PK-resistant core remains. It's insoluble in detergent. These are all features of an amyloid protein. As I mentioned in the previous slide, this is the infectious agent. And then until a couple of years ago, I could just say, well, it had beta sheet structure, but now we know through cryo-EM studies that it has a parallel and registered intermolecular beta sheet structure. So we have a very high detailed structure of the infectious conformation of the prion protein. So in the field, and I'll catch myself slipping to this once in a while too, it's referred to as prion replication. And I think replication is a poor way to think about this. I think conversion conceptually just, I think, makes a lot more sense. And what's thought to happen is pictured here in this little blue fibril is that this amyloid self-templating PRPSC will interact with the normal form of PRPC. It will bind. And then through a process that we have zero idea how it works, is that somehow PRPSC will convert the conformation of C to SC, therefore making more PRPSC. In the cell is that these fibrils will grow and then they'll be fragmented. And since the conversion occurs on the free ends of these fibrils, you can see that this results in this kind of self-feeding loop that results in exponential conversion or exponential replication of prion infectivity. And the mechanism I think that best describes this is nucleation-dependent polymerization. In this model, these are all examples in closed systems. So if you have your monomeric protein and your fibular or your prion form of the protein, is that you have a lag time, formation nucleus. Once the nucleus forms, you reach this kind of exponential phase, and then you plateau where you get an equilibrium between the prion form and the monomeric form. This lag time, pictured down here, this is thermodynamically unfavorable, and it's also related to the concentration of the monomer. So the higher concentration of the monomer, the shorter the lag time, and vice versa. And then once this nucleus is formed, now thermodynamically, the C to growth becomes very favorable, hence the exponential form of the fibril form. If you take this preformed fibril and you add it into a tube of monomers, you basically skip this lag time and you go right to the exponential or logarithmic form of prion formation. And as I'll talk about in a few slides later, this model very nicely explains the various etiologies of prion diseases. So some of the physical properties of prions are really unusual. Again, you have this protein-only infectious agent. It's an amyloid fibril. And prions are not an underlined not, so they're not inactivated, as I mentioned before, from Tika's work, ionizing radiation, UV irradiation. So you can boil this stuff, it doesn't care. Nucleases, doesn't nucleic acid genome, okay, so it doesn't care. Proteases, it is highly resistant to proteolytic degradation. You can treat this material with high concentrations of PK for literally days, and it still remains. Formalin, and I'll show an example of this in a few slides. You can formalin fix this material, and it's still infectious. And then probably most concerningly is autoclaving. And here I'm very specific, and this is 121 degrees C for 20 minutes. This is kind of standard autoclaving conditions in microbiology departments and clinical labs, is that it survives autoclaving. And just kind of a few examples of this. This is work from Paul Brown's group back in 2000. And what he did is he took prion-infected brain material treated at various temperatures. I'm just going to focus on the 600 degrees C, where you take this material, you put it at 600 degrees C for 15 minutes. It actually catches on fire and smoke rolls out of the crucible. And you have this black ash in the bottom of the crucible. Put a little bit of PBS in, resuspend the black ash, you inoculate it in animals, and you see 5 out of the 18 animals develop prion disease. Okay, tough stuff. As I mentioned, it survives formalin fixation, and this has been known for a long time. So this is work from the 1970s. And they had TME. This is and I'll talk about this in a minute. It's a prion disease of mink, and you can let TME brain sit, basically for years in formalin or in paraffin tissue blocks for several years, and the materials, it's still infectious. And actually, this is something we use in my lab all the time, is that we can go back to like H&E slides, pop the cover slip off, and we can recover infectivity out of there and do characterization. So it survives formalin quite readily. So you might be thinking, it's like, how do we get rid of this stuff? So a couple of things is bleach, sodium hydroxide with various combinations here. These are all recommended by the WHO on how to inactivate prions. And then autoclaving, but very specifically at 134 degrees for 18 minutes. And this is what we use in our group. But just be aware that autoclave bags are not rated for this. You have to buy special ones. So if you're going to do this unknowingly, you have just a big puddle of plastic in the bottom of the autoclave. Don't ask me how I know that. All right, so prion strain diversity. So again, you might be wondering, you have this protein-only infectious agent. Is we know that there are prion strains. And prion strains are operationally defined as a heritable phenotype of disease under a fixed set of agent or host parameters. And the phenotype of disease can include clinical signs. So in the same host species, you can have 2 strains of prions that have distinct clinical features of disease, distinct incubation periods of disease, distinct neuropathology. So pictured here is the distribution of spongiform degeneration in 2 prion-adapted hamster strains. They're very different. The agent distribution, so pure PSC, can not only pictured here on the right differ within a tissue, this is brain, but can also differ between tissues. So tropism can differ in prions. And then lastly, and this is very important for chronic wasting disease, is the zoonotic potential. And that is different prion strains in the same host species can have different abilities to infect a new species. So just the mere existence of prion strains was actually probably the best evidence against the prion hypothesis. And this is what proponents of the viral hypothesis used to support that prions are caused by a virus, because, well, how do you have this information encoded without a nucleic acid genome? And so lots of work has really kind of added to this idea that distinct conformations of pure PSC encode strain properties, and that the informational molecule is that of conformation. And so indirectly, this was noted by differences in migration in STS page, pictured here, relative resistance to PK digestion, and conformational stability, which is basically, you go back to your, think about biochemistry, it's a melting assay. So indirectly, there's evidence to suggest that the pure PSC molecule from different strains has these different properties. And then recently, with the advent of cryo-EM and being able to solve the structures of pure PSC from different strains, indeed, cryo-EM structures have revealed that they do have different conformations of pure PSC. But I think what's really important to realize here is that we still don't understand the structure-function relationship between how these distinct conformations encode these really complicated biological properties that prions have. All right. So talked a little bit about prion agent. Again, this infectious only and protein only infectious agent. It can encode strains. And now what about the diseases that they cause? So prions cause a wide range of diseases in animals, including humans. And in humans, prion diseases are comprised of Kuru, or the Fori people of Papua New Guinea, Gerstmann-Strasser syndrome, fatal familial insomnia. These are both familial prion diseases. And then CJD, or Creutzfeldt-Jakob disease, which is unique in biology in that it has 3 distinct etiologies. It can be transmitted by an infectious agent. It can have a familial etiology and also a sporadic form of the disease, which actually is about 90% of all cases of CJD. Again, something that was really difficult to understand is how can you have a sporadic disease In cases of sporadic CJD, you can take brain material from these patients and transmit it to non-human primates. Very difficult for kind of a viral etiology to explain. But as I mentioned before, on how prions form, the prion hypothesis provides a very clear mechanism of how you can have this de novo creation of infectivity. In animals, prions are comprised of scrapie of sheep and goats, transmissible and mink encephalopathy of ranch-raised mink, bovine spongiform encephalopathy or mad cow disease in cattle. The most recently identified prion disease is camel prion disease. There's one paper on this. And then chronic wasting disease, which I'll talk about in more detail for the rest of the talk. So CWD occurs in captive and free-ranging cervids. It's in whitetail deer, mule deer, elk, moose, reindeer. Pictured here in a map of North America just gives some dates when CWD was first detected in either captive or wild populations. Pictured here on the bottom is a graph just showing the number of states that are CWD positive. Starting back in the 70s and then really kind of taking off in the mid-90s. And you can see there's a large growing number. And this is the current distribution of CWD in North America. And right now we're up to 32 states that currently have CWD. Pictured here in the dark gray is where CWD was first identified. And you can see it's spread throughout most of North America. It's also been identified in South Korea, and this was from importation of elk from North America that were subclinical for CWD. And then most recently, CWD has also been identified in Nordic countries, in moose, reindeer, and red deer. And as I'll talk about kind of later, importantly is that the form of CWD in Nordic countries is very distinct from what's occurring in North America. In the areas where CWD exists, it can have it's an increasing incidence. And so, for example, there are areas in Alberta and in Colorado where CWD, sorry, my lights go off, where CWD can have an incidence of over 60% of the animals that are tested will test positive for chronic wasting disease. So you have these really high incidence of disease, which can affect herd health. The host range of CWD is poorly characterized. There's controversial, kind of conflicting evidence of transmission of CWD to non-human primates. There's been a lot of work trying to transmit CWDs to sympatric species. And here's an example is that CWD can be transmitted to swine, which are sympatric with deer in North America. There are numerous strains of CWDs. I mentioned kind of in the introduction to the prion agent is that strains can have different zoonotic potential. And this is one of the concerns in the field is that there are beginning to identify different strains of CWD. And we don't really kind of fully understand the consequences of that. And then one of the most challenging things about CWD, and I'll talk a little bit more about this in another talk, is that it can persist in the environment. So it's been known for a long time, and this is with sheep with scrapie, is that it can persist in the environment for here at least 16 years, again, to the unusual protein-only nature of the infectious agent. And it's been shown that prions can bind to the soil and remain infectious. They can be taken up by plants and grass. And then our group is really interested in prion evolution. And something that we're finding is that the environment can actually be a mechanism of helping prions evolve in that certain strains of prions will either interact with soil particles better or worse and survive weathering differentially. So we think that different soil conditions and different weathering patterns can influence the strain distribution of prions in the environment. Okay, so this brief kind of introduction to the prion diseases, I think it's important that we kind of understand that this agent is really kind of unusual, and it's going to, I think, help us when we get into CWD understand some of these really kind of crazy results that I'm going to talk about. And what I want to do is kind of give this historical overview of the fields, how they're really interrelated and build upon each other. And I'm going to start in the 1980s, because at least for CWD, this is a really clear start point. And really the founder of the field is Beth Williams. And Dr. Williams was a wildlife biologist at Colorado, and she identified the spongiform encephalopathy of deer. Pictured here in figure 1, this is a female mule deer, and you can see it's emaciated in a wasted state, has excessive salivating, and the neuropathology of animals what she described was consistent with the spongiform encephalopathy. So pictured here, you can see these kind of white vacuoles. This is spongiform, looks like a sponge, okay? And then encephalopathy, all that means is it's neurodegeneration in the absence of an inflammatory response. So the neuropathology was consistent with this group of diseases. And in this seminal paper from Dr. Williams identified 53 cases in mule deer, 1 black-tailed deer in captive facilities in Colorado and in the Front Range in Wyoming and around Laramie. So other work coming out of the 1980s, again, from Beth Williams, who's the founder of the field. She identified this in captive elk. So again, the spongiform encephalopathy of Rocky Mountain elk. The neuropathology of CWD is characterized by these large florid amyloid plaques, which is also seen in a human prion disease. So Carlton Gajusek, who won the Nobel Prize for his work in Kuru, investigated the neuropathology of CWDs because it was reminiscent of human prion disease. And then something I just want to point out here in the early 1990s, just the challenges that the early investigators faced is that there's these kind of unpublished reports of CWD in free-ranging deer. Prior to this, everything had been in captive facilities. And kind of why that is because their ability to diagnose CWD was really based on H&E sections of brain. So this required necropsy soon after death. So I think it's pretty remarkable that they're able to identify any cases in wild deer kind of based on just the difficulty of collecting tissue and getting this fixed and processed in a time that it wasn't degraded. And as I go through the decades here, I just want you to appreciate just how sophisticated our diagnostics capabilities for prions has really become. As I mentioned in the introduction, again, this protein-only infectious agent. The real kind of nidus of this started in the 1980s again, and at least from my point of view, there are 3 papers that are really kind of the pillar of the prion hypothesis. The first one was published in Science by Stan Prusner, and this is the idea that there could be this protein-only infectious agent, and kind of gave this mechanism of how it worked. The second one is, again, from Prusner's group, published in Science. David Bolton, who's a postdoc in his lab, identified the protein that purifies with infectivity. And this was a Herculean and just tremendously difficult experiment. Picture here, this is not a Western blot. This is total radiolabeled protein from prion-infected rodents. And they would then assay, biochemically purify infectivity, and then ask what proteins were present. And what they found is, here the S is for scrapie and the N is for normal, is identified this protein that co-purified with infectivity, that resisted PK digestion because they knew from previous studies that occurred. And so now they have this candidate protein for this idea that Prusner proposed. But it's also possible, well, this is a viral protein. It's not really a prion. And then a few years later, from Charles Weissman's group, published a paper in Cell. And again, this is in the 1980s. So now this is really super easy, but back then this was very, very difficult. And what they showed is that this protein, the mRNA, was present in both uninfected and infected. So it was a host-encoded protein. So these 3 papers really kind of built the kind of bedrock of the prion field. And at the time, it was very, very controversial. Lots of yelling at meetings. So in the 1990s in CWD, really kind of focused on pathogenesis. And again, the seminal studies in pathogenesis were performed by Christina Sigurdsson, who was a graduate student in Ed Hoover's group at CSU. And what Christina did is did oral transmission studies of deer and did a temporal spatial studies to follow the spread of PRPSC. And in this first paper focuses on outside of the central nervous system, mainly in lymphoreticulaire system tissues. And one of the things that really struck me reading this as a graduate student at the time is that these are diseases of long incubation periods. Another name for the diseases at the time were slow viruses. But what Christina found is that she could find PRPSC in lymphoid follicles at 42 days post-infection, which was a really remarkable finding and really kind of indicated that things happen much more quickly than had previously been appreciated. Also, this retropharyngeal lymph node was the first place that prions were detected following oral infection. And all of our diagnostic efforts to date are based on this observation because this is the tissue that is collected since it's the first one that becomes prion positive. Another paper from Christina that she published following up on this, again, showed the temporal and spatial spread of PRPSC in the nervous system. And this basically mapped out how prions spread following oral infection. It followed known neuroanatomical routes of infectivity, and then subsequent studies looking at wild deer basically were able to recapitulate the findings that Dr. Sigurdsson found when she was at CSU. So some very important initial studies on CWD. Also in the 1990s, Beth Williams mapped out the neuropathology in fine detail in various regions of the CNS. And one of the interesting things that really wasn't realized at the time is that there was variation between groups of animals, which probably is some of the first indication of distinct strains. Other work that came out in the 1990s that were super important was from here with Catherine O'Rourke. And Dr. O'Rourke did a bunch of work in sheep scrapie prior to moving to CWD, and she identified that in sheep, there are polymorphisms of PRP that really correlate with susceptibility to infection. So now when she looked at CWD infected animals, she found something very similar. And here's that there's a polymorphism at 132 in the prion protein of deer that seems to influence susceptibility. And this was kind of the start of this whole subfield in CWD, showing that PRP genetics has a large influence on susceptibility to deer to prion infection. So in prion diseases in the 1990s, two things I wanted to point out. The first one is from Stan Prusner's group, and this is the use of transgenic animals. In the rest of the talk, these are used extensively. This has been a super important model system, not only in prion diseases, but lots of other areas of infectious disease. And what this study showed, not only is could you recapitulate prion disease in these transgenic animals, but that the prion protein basically entirely governed the species barrier effect or susceptibility of prions going from one species to another. It was known that mice were very resistant to infection with hamster prions, but now if you make transgenic mice that express hamster PRP, they become totally susceptible to hamster prions. So a very, again, seminal observation that will be built upon in the subsequent decades. I've already shown this Western blot before. This is from Richard Besson and Dick Marsh at the University of Wisconsin. And they were really the first to show that distinct conformations of PRPSC or propose this idea of the conformational idea of prion strain diversity. All right, chronic wasting disease in the 2000s. So now building upon this transgenic mouse technology, Glenn Telling's group at CSU is they made transgenic mice that expressed Cervid PRP. Again, it was known at the time as that wild type mice were fairly resistant to infection with CWD, but now when you introduce the Cervid transgene into these mice, they become susceptible to infection with CWD. So again, showing the importance of PRP in the species barrier effect, but also these animals have PRPSC, they have neuropathology, totally recapitulating the disease in these mice is what we see in deer. And these have been a super important tool for understanding prion pathogenesis and then also detecting prions. Again, building upon this idea of the prion protein being the main mediator of the species barrier effect, Periluigi Gambetti at Case Western, what they did is they built transgenic mice that not only express Cervid, but also human PRP. And what they found is that if you have transgenic mice that express elk PRP, they're susceptible to elk prions, okay, consistent with Telling's group. Now they make transgenic mice that express human prions or human PRP, they're susceptible to human prions. And now they ask, if you inoculate these humanized mice with CWD prions, what happens? And they wait 600 and 700 some days and they don't get sick. So indicating a very large species barrier may exist. Again, a whole subfield in CWD research kind of based on this. There's lots of papers. Most recently, there's a paper from Canada suggesting that there is evidence for transmission of CWD to some of these humanized mice, but it may be at very low levels. In the 2000s, this is work from Joel Peterson, again, University of Wisconsin. And what Peterson's group was the first to show is that we knew with sheep scrapie, and we had kind of some evidence with CWD, is that it could persist in the environment, but we didn't know how or kind of where. And what Peterson's group showed is that prions could bind to soil. And interestingly, what he found is that there was a kind of a specificity, if you will, is that clay, like clay materials, would bind prions more avidly compared to sand. But then importantly, when you bioassay this material, is even when prions are bound to these surfaces, they remain infectious. And so this was really kind of the start of the whole environmental prion field, mainly in CWD. Numerous papers, and this is, our group has been very involved in this work, trying to understand how do prions interact with these surfaces? What are the mechanisms of binding? How does it remain infectious? How does it respond to weathering? Are there strain-specific differences to CWD agents, based on environmental conditions? So a very important kind of seminal observation back in the 2000s. Again, that's been followed up by, and this is just a handful of papers that have followed this up. In the 2000s, again, building on the work of the transgenic mouse studies, and then also Christina Sigurdsson's pathogenesis studies, is what was found is that deer infected with CWD shed prions into the environment, and they can do this numerous ways. And this first paper from Stan Prusner's group is what they found is that infectious prions were present in feces. This was from mule deer, basically the entire time course of disease. Which can last several years. Glenn Telling's group, again using transgenic mice as the assay, found that skeletal muscle of deer infected with chronic wasting disease contains infectivity. So this is important for species, including our own, that consume skeletal muscle. And then lastly here, I wanted to point out, and just because this I think was a paradigm-changing observation, is that Candace Mathiasen in Ed Hoover's group found that prions were in saliva. And again, might provide a mechanism of deer-to-deer transmission. But I think more importantly, it showed that prions were detected in blood. And subsequent work from this group has shown that there's this long-lasting prionemia in blood of CWD-infected deer, but also in other prion-infected animals. And at the time, pathogenesis, kind of the paradigm of pathogenesis was infection of spleen and lymph nodes, infection of peripheral nervous system, infection of central nervous system, then death of the host. And this work really showed it was more complicated than that, is that you have the involvement of prions in blood, and we're still trying to figure out kind of the significance of this on how prions spread throughout the animal. In human prion diseases, this is also this is a very, very significant observation as well. So this occurred back in the 2000s, and there's again a whole series of literature about investigating prionemia. In the 2000s, in prion diseases, there were two techniques that, in my opinion, really revolutionized the field. So if you think about how do you detect infectious agents? So it's usually an antibody against the infectious agent. You're detecting the nucleic acid genome via PCR or something like that. In prion diseases, there's no nucleic acid genome. There's no immune response to the agent, and you just have this protein. So everything up to this date was Western blots, IHC, which are great, but they're not very high sensitivity. So what Claudio Soto's group did is develop this technique called protein misfolding cyclic amplification, or PMCA, and conceptually think about this as PCR, but for misfolded proteins. And what you do is you take a small amount of this misfolded seed and you put it in excess of pure PC, which at the time was from a brain homogenate from a rodent. You get the prion conversion occurring in the test tube. Here you use sonication to shear the growing fibril and making smaller fibrils, again, these free ends that can feed into this self-amplification loop. And what they found is that they could not only produce basically an infinite amount of PRPSC, but that it was highly sensitive. So very, very low amounts of PRPSC could be amplified up, analogous to PCR, to levels that could be detected by conventional techniques. So importantly, this is bona fide prion formation in that this material is highly infectious. And because it's actual prion formation occurring in this test tube, it recapitulates a lot of the biological features that we're interested in. Species barrier effect, strain properties, anti-prion drugs, all can be tested with this biochemical technique. So a very revolutionary technique. So another revolutionary technique that came out, and this was from Byron Coey's group at NIH, is called RT-Quick. And it's kind of the same, but it's a little bit different. It's the same in that it's again an in vitro technique. Here you're using recombinant PRP, and then your detection method, instead of being a traditional amino assay, you're using a thioflavin T, which binds to beta sheet structure proteins and has a change in its fluorescent properties that you can measure on a plate reader. And through a series of papers kind of refining this technique, now RT-QuIC again, this is on the kind of the prion field, this is very rapid. So in about 2 days, you can get results and you can detect basically less than an LD50. So super sensitive, very rapid, relatively easy to get set up, has high sensitivity for detection. Importantly, this material is not infectious. So unlike PMCA, which recapitulates prion formation, this does not. This is a detection technique. And since this reaction material is not infectious, from a diagnostic lab point of view, this is actually really a good thing. And this RT-QuIC right now is being integrated in by the USDA for their detection methodologies for CWD. And it's also been, and this is kind of off topic for today's talk, but you can do this with other protein misfolding diseases. So there's lots of groups using this technique to develop preclinical tests for Alzheimer's disease, Parkinson's disease, et cetera, using this methodology. So very, very powerful methodologies, which, as I'll show here, are now used in CWD research. So in the 2010s, a huge experiment at the NIH. So this is at Rocky Mountain Labs in Hamilton, Montana. Brent Race, Bruce Cheesebro, and what they did is they inoculated non-human primates with CWD and waited basically 13 years and asked, do they come down with disease? And the short answer is no. There was no clinical disease, no neuropathology. Again, using this new technique, RT-QuIC, they could not detect residual prion infectivity in these animals. There wasn't a subclinical infection going on. And so this is, again, a very, very important observation in the field. This is in contrast, and this is from Justin Greenlee's group at the USDA. This work was done in Ames, Iowa. Again, where they inoculate the natural, not the natural host, but the real host, this is pigs, swine, with CWD, and ask at market weight or at later time points post-infection, is there disease? And the kind of long story short is that they get a subclinical infection. Again, using these newly developed techniques to assay for the subclinical infection. And this is really an important observation in that pigs are sympatric with CWD. And if they become subclinically infected, this poses the risk that this could adapt to this other species. And then other work that's come out of Greenlee's group since then, that I think is really important, is that they were able to transmit CWD into sheep, another sympatric species with deer. And then they found that if you passage CWD into metavoles, again, a sympatric species with deer, is that it changes the nature of the CWD agent and now becomes more pathogenic for raccoons. So why is that important? Is because back here, all of these studies are directly asking the question, can CWD from cervids cause disease in non-human primates? But if CWD gets into a sympatric species, it can change the agent. And now what we know about the host range of CWD, we have to reevaluate. In the 2010s, Claudio Soto identified prion uptake in plants directly from infected soil. These plants, of course, die and go back into the soil, but it raises the possibility that prions can be transmitted via plants. And then work from Glenn Telling's group when CWD identified a brand new mechanism of how prions can be transmitted to a new host species. And this is called non-adaptive. With most infectious agents, including prions, if it goes from one species to another, you have a species barrier effect, but then subsequent passage results in a shortening of incubation period as more pathogenic. And what Telling's group found is that you can have instances where it doesn't adapt to a new species, but retains infectivity for the original species. So conceptually, a very, very important idea that was founded in CWD. In prion diseases in the 2010s, this is where synthetic biology came into vogue. And so work from Jian Ma, basically taking all recombinant proteins and totally in vitro you're able to de novo generate infectivity. So in the green are negative controls, and 2 and 4 are the experimental groups, and you can, I'm sorry, 4 is with PRP, and you can in vitro generate prion infectivity. And our group has done a lot of work with synthetic prions as well, and we can show we can model interspecies transmission using this synthetic technology. And then probably most surprisingly to me, at least, was work from Charles Weissman's group showing that you could treat prions with anti-prion drugs and actually get drug-resistant prions. And if you remove the anti-prion drug, is that the prions would then rapidly revert back to the drug-sensitive state. And kind of based on this work, Weissman, who early in his career worked on viral quasi-species, it's like, oh, prions also behave as a quasi-species in the sense that they can rapidly adapt to these new replicative environments. So again, a very surprising kind of observation for a protein-only infectious agent. And then in the present, the biggest news with CWD is, again, this identification of CWD in Nordic countries. There's been a series of papers. And in Nordic countries right now, they are actively trying to manage this disease before it kind of gets out of control. And importantly, strain typing of the prions in CWD infected deer in Nordic countries is very different from what we observe in North America. Also in CWD, again, numerous studies that are starting to identify more and more different strains of CWD. And again, the concern about this is that different strains can have different zoonotic potentials. So this is something that we're very concerned about and trying to address. And then another thing that again, using transgenic mouse technology and using this RT-Quick technology, and then also building upon Catherine O'Rourke's work, showing that PRP polymorphisms affect susceptibility, is that there are certain polymorphisms of cervid PRP that are fairly resistant to infection. But what they're finding is that while they don't develop clinical diseases, they're also, they don't develop clinical disease, but they still are replicating or converting agent. They're still a shedding agent. So from a management point of view, this is very important to consider if you're going to breed deer to have a certain polymorphism. It's just, what does that really mean? And then at present, the biggest thing in prion disease against cryo-EM, there's tons of groups now working on cryo-EM. The first one came from Alison Krauss, and she was the first one to solve the structure of PRPSC, picture down here. Some studies that came out about a year later out of the UK, again, solved PRPSC from these distinct strains, as I mentioned in the introduction to the talk. So the most direct evidence to date that strains are encoded by distinct conformations of PRPSC. And so kind of summarizing kind of, from 1980 to the present, is that in the prion field, you had these really huge challenges. Is that in the 1980s, the prion agent was very controversial and it had these unusual properties that you really couldn't understand or explain with a viral etiology. So you had the prion hypothesis, okay? Totally unconventional way to try to identify or describe an infectious agent. Turned out to be right. Again, a protein-only infectious agent, you have strain diversity, distinct conformations, a brand new way of thinking about how to encode information. Large animal bioassay. In the early days, things had to be done in deer, which was very expensive and difficult to do. Now you have transgenic mice, PMCA. I didn't have time to talk, but there's cell assays for prions as well. Again, this whole problem of how do you detect prions with just a protein-only agent. So you come up with these really innovative techniques, PMCA, RT-Quick. And then what my group is really interested in is, again, how does this protein-only agent evolve? How does it respond to new replicative environments? And Charles Weissman, quasi-species, Napa, our group has done a lot of work on strain interference and evolution as well. So you have these really unusual challenges, which at the time seemed to be kind of insurmountable, but over the decades, they've all been solved. And so with CWD, there's some really significant challenges, is that you have this increasing geographical range of disease, unknown strain diversity, kind of a poorly characterized zoonotic potential of the agent, kind of which is complicated by strain diversity. It can persist in the environment, which means that mitigation is going to be very difficult since this is a disease of wildlife. And this leaves really few management options for our wildlife biologists to try to mitigate disease. In spite of all these challenges, I'm actually really optimistic about the field. And part of this is really outstanding work that the trainees are doing right now. So I just got back from an international CWD meeting, and here are a few abstracts showing for detection methods. Now groups are actually working on field portable detection methods, kind of modifying RT-Quick. There's some really cool work from our wildlife biologists and behavioral specialists where they're trying to use adaptive machine learning and GPS data to actually predict where CWD is going to occur based on deer behavior. There's lots of work on actually doing detection of CWD in environmental samples, which has been really challenging, but is needed to help monitor where disease is at. In pathogenesis and transmission, much more work has been happening, understanding kind of the role of blood in CWD. Also some really exciting work showing that mother to offspring transmission can occur. So knowing this is really important for mitigation of CWD. And then also some work showing that invertebrates may actually play a role in transmission of CWD between deer. In therapeutics, now that we have the structure, PRPSC groups are working on developing antibodies to the structure for vaccines. And so there's active vaccine development that's occurring. Kind of concerningly, as I mentioned, with swine being sympatric, now people are starting to look at feral hogs, and there's some evidence that there actually might be CWD in these animals. So that's of concern. And then with prion strains, a lot of work that's occurring in Norway. And what's super interesting with this, it suggests that some of the CWD in Norway is spontaneous, and when you passage it, it evolves and becomes and looks more like what North America looks like now. So people are speculating that in the Nordic countries, this may have been North America back in the 40s or 50s. So what I've hoped to convey, over the short period of time is that the prions are this unique infectious agent, comprised solely of protein, have some really weird properties because of it, that cause lots of different diseases in animals, including us. And then specifically for CWD, this was first started by Beth Williams, or first identified by Beth Williams in the 1980s. And through a large, collection of work by both people working in CWD and the prion field, kind of together, had really made tremendous amount of progress in the field. So with that, I'd like to thank the ASM for this opportunity. I'm very grateful to be part of this. I'd like to thank the people in my lab and all of my colleagues, both in North America and worldwide, that I have the pleasure of working with, the various prion groups that I'm associated with, the funding agencies. And with that, I'd be happy to address any of your questions. Thank you.
Thank you so much. This was really fascinating to hear. As someone who had to write a paper about the prion hypothesis and take sides in a discussion that was in fact very heated back in graduate school many years, decades ago, this was fun to see all of the newer research as well. And we have a lot of questions in the chat, so we'll try to get through as many as we can. And for those of you who have already entered your questions into the chat, thank you very much for doing so. I will do my best to pronounce names, but I apologize if I don't get them all correct. So I'm going to start with James Misselli from UMass Dartmouth. Actually has asked a few questions in here, and this the first one I think you did answer later in the talk about whether interactions between PRPSC and PRPC occur at specific amino acid sites. But the follow-up to that was, does it differ in differing strains?
Yeah, so there's a few papers suggesting which amino acids of PRPC interact with PRPSC. But people have not, they've only done this with one strain. So I don't know if different strains would have to, I would presume that different strains would have different interactions. But it's unknown.
And the next question also came from James. Was there a line ID'd of a species barrier? If so, is it all non-human primate barriers or only human? I guess, can you speak a little bit to what is known about potential species barriers?
Yeah. So the species barrier is almost entirely dictated by the prion protein. And so we know that differences in the amino acid sequence between PRPSC and PRPC will influence the species barrier. But right now we don't have any method to actually kind of predict that. And I think it's because, so part of it is just we don't have, well, we're just starting to get good structural data on PRPSC, and we have some structural data on PRPC. I mean, you can map these, you can make transgenic mice where you substitute amino acids and show that you can either increase or reduce the species barrier effect, but it seems to be very specific just for that combination. We haven't been able to make kind of universal, it's like, oh, well, it's this region. And so you can make some better predictions with, for example, humans and CWD.
So there were two that came next, both related to questions regarding immune system. So Ikra Mukadas from Rawalpindi Medical University in Pakistan asks about prions entering our bodies or entering hosts. Are there mechanisms evolving for immune system escape? Could you speak to that?
Yeah, so interestingly, I mean, the immune system is basically completely blind to prion infection. So, so the only kind of interaction that prions really have with the immune system is that there's some evidence that dendritic cells may transport them from the gut to draining lymph nodes, but it's more of a kind of a Trojan horse kind of mechanism. And then also prions will convert and accumulate to high levels on follicular dendritic cells in spleen and lymph nodes. But there's absolutely no kind of phenotype or pathology because of that.
So presumably then we already have the answer to the next question. Will Horn had asked who's a recent graduate from North Arizona University, had asked if there's any immune system evolution against prion diseases.
Not that I'm aware of.
So Roa Rashad from Alexandria University Faculty of Science asks if prions are considered as living particles, either when associated with living cells or not.
Yeah, that's a great question, and that's a philosophical question, I think. Yeah, it's an intracellular obligate parasite, just like a virus. Whether that's alive or not, I guess yeah, that's philosophy, not biology, in my opinion.
Well, so I'll ask a partial follow-up on that. Since you told us about some of the ways in which the infectious particles can exist out in the environment on surfaces like clay, so are they considered in that context as well then?
Well, I mean, so they're considered to be infectious out in the environment for decades. Yeah. Yeah, and they can survive. I mean, work that we've done has shown that you can bind prions to clay and make this, basically prion dust. And if you dehydrate it, this stuff is incredibly resistant to weathering and other forms of inactivation, and could presumably persist, for decades easily.
And can the conversion or sort of increase in the infectious particle size grow outside of a host cell?
Not that we're aware. It requires PRPC, and PRPC, like normal proteins, once it's in the environment, is destroyed very rapidly.
So there's the obligate intracellular component.
Correct.
Great.
Yeah.
Okay. So next question came again from James Maselli. Assuming ideal PRPSC binding conditions, are there factors that mitigate infectivity?
So yeah, so there is a series of, or a class of anti-prion drugs that are known to bind amyloid proteins. So some of them, like Congo red, are a histological stain for amyloid. And what happens is that Congo red molecules will intercalate into the beta sheet structure of PRPSC, and it will overstabilize the structure. So during that either binding process or the conversion of C to SC, either it's physically blocking the binding, or probably more likely is that PRPSC has to be somewhat flexible to get the conversion process to happen. And then binding of that dye to PRPSC overstabilizes it, preventing that flexibility, preventing conversion.
Damilare Jugbe at Oklahoma State University asks about anti-prion drugs. So are there such inhibitors? And that again is partially related to the last question, and wondering how all of this translates to development of drug-resistant prions.
Yeah, so I think one of the unique things about the prion field is so there's a whole line, I mean, there's a whole big series of papers on anti-prion drugs with different mechanisms, but you always kind of get this, I mean, the same thing in in any kind of microbiology is drug resistance is always a problem. So I think so if our lab was going to say, we are going to work on anti-prion therapies, I would not work on anti-prion drugs because of the drug resistant issue. Where prions are different compared to, let's say, viruses, is I can actually now target the host normal form of the protein. And that's what groups are doing right now, is they're asking, can we downregulate that protein? Or we know that, so when I talked about the transgenic mouse technologies, so if you make a knockout mouse, if you knock out the prion protein, these mice are totally resistant. They don't replicate agent, they don't develop disease because you remove the template for conversion. And in fact, at the USDA and Ames, they made a knockout cow. So they took a cow and knocked out the prion protein. The cow is resistant to mad cow disease. You know, I've seen the cows, I mean, it looks like a cow. And so I think that, you know, more maybe in agriculture settings, you might have kind of more genetic means, but in human settings is either trying to figure out ways to downregulate the gene or figure out therapeutic drugs that will, I don't want to get into the weeds of the cell biology, but there's certain forms of the normal form of PRPC that are less convertible. And can you treat an individual with something that drives PRPC to either a location or a conformation that will prevent prion formation? I think that's a much more viable mechanism or means of anti-prion therapeutics compared to anti-prion drugs. Because I think with any anti-prion drug, eventually you're going to develop resistance.
We have a question from Boston University School of Medicine. Nadia Misra asks, this was related to earlier in the talk when you mentioned the different geographic locations for CWD. Do you see different deposition of PRPSC in the Nordic versus North American CWD?
Yeah, so the biochemical properties of PRPSC from some of the Nordic animals is very distinct from what we see in North America. In moose, in the Nordic countries, PRPSC is restricted to the central nervous system. And so people are speculating that this represents a sporadic form of disease, which, again, as I mentioned, occurs in humans, and which I didn't mention can occur in cattle. So there's very, very rare forms of bovine spongiform encephalopathy that occur in aged cattle, mainly dairy cattle, that are atypical and we think are actually spontaneous.
Right. I'm going to read out two remaining questions in the interest of time, but I think these two are really great and I want to make sure we get to them. So we have one from a master's biology student from Diné College, Faye Beke, who asks, is there any information on sheep in the Navajo reservation having prions, being that sheep meat is very popular among the Navajo people?
Yeah, so specifically that I don't know, but what we do know is that sheep scrapie has been around for hundreds of years. I mean, there's actually pretty good evidence that existed in the UK 250 years ago, and there are countries that have incredibly high rates of scrapie in sheep. those countries have the same incidence of human prion diseases compared to, let's say, like Australia and New Zealand, which have remarkably low levels of scrapie. So there's very little evidence that scrapie can be transmitted from sheep to humans. There's really good evidence that mad cow disease transmitted from cattle to humans. And kind of the question with CWD is, where does it fall along that spectrum? That's what we're trying to figure out.
I think this is a great question to end on. Also, again, from Damilare Ajagbe, do prion proteins have other functions that might be beneficial?
Now, great question. And when the PRP knockouts were first being proposed, there was kind of a side betting in the field, if you will, what the phenotype would be. And almost everyone said it was going to be an embryonic lethal because they're very highly conserved across species. And it turns out there's hardly any phenotype. And it's been very difficult to pin down a function of PRP-C. Now, having said that, and I didn't have time to talk about this at all, there's a whole other field of prions in yeast. And it's totally different proteins, but it's a prion in the sense of it is a protein with one conformation that can transfer that conformation to another protein. And in yeast, that gives it a different phenotype. So yeast biologists think about prions as an epigenetic means of inheritance. And in that system, yeah, prions are actually useful.
Great. Well, we've gone well over our hour, thanks to all the wonderful questions from the audience. And so we're going to wrap up now to be mindful of everyone's time. So first, I want to thank Dr. Bartz again. This was a really fantastic presentation, and thank you for staying on a little extra to take so many questions. I also want to extend a thank you to Kirsten Bartlett and Ann Dempsey from ASM for organizing this ASM-sponsored series. And again, thank you to all of you for coming and for really excellent questions. We encourage chapter and branch members who were not able to view this to listen to the recorded seminar, and that will be made available for participants who've registered after the event. If you have any questions, you can email the student chapters at asmusa.org. I think Kirsten will put that into the chat for you. And we wish you all a great day. Thank you so much.